Here we move into the physiology of the pump component of the circulatory system. And in humans, that is the heart, or as an adjective, we can call things cardiac, whatever. So cardiac physiology. And this is going to be the first of two segments that I'm going to do on cardiac physiology. So really, in case you weren't familiar, the pump of our circulatory system is the heart, and we have a chamber type heart. There are some other types of hearts that are peristaltic type hearts that essentially pump blood the same way that your intestines move chyme through your intestines. We don't have one that works that way. We have a chamber type. That essentially you have this hollow area that is going to fill up with the fluid portion or the blood, and then is going to contract to squeeze that fluid out and propel that through the vascular system. And we have one-way valves generally in these chambers that are going to ensure that when we squeeze the fluid out of this chamber, it's going to go in the forward direction, the direction that we want it to go, and not back where it just came from. So essentially we have a heart that's made up of these chambers, and that are going to squeeze fluid out and do this through one-way valves that are going to ensure which way that's going. We have a couple different chamber types that we need to talk about when we talk about the heart. And the first one is an atrium. An atrium is just a smaller, less muscular chamber that is essentially receiving the incoming blood. And we also have ventricles that are going to provide the force for the outgoing blood. So ventricles tend to be larger and more muscular than atria are. And in mammals, we actually have essentially a dual pump design. Your heart, while being one organ, is really two different pumps acting in unison, pushing blood out to two different directions. And that's because we have a circulatory system that is dual circuit. So if you weren't familiar with this, let's go ahead and just review this really quick with this simple diagram. Here we have a heart here, and we have two different circuits that go through the body, or go through our circulatory system. One of them is going to push blood out of the heart down here to your lower right, and it's going to push it out to the body here. And so that's nice because we can push this out into capillary beds, which is what this is representing here. Pushing it out, we have a lot of oxygen in there that's going to be used up by active working tissues, and it's going to be returned back to the heart after that. Now, one thing that's interesting is this first portion is going to be high pressure. But after you pass blood through a capillary bed, there's a lot of resistance in a capillary bed because we have many, many small vessels. And you have a lot of surface area that's in contact with your fluid. And so this builds up a lot of resistance. And so what happens is, after it comes out this capillary bed, we're at low pressure now. So we need to push this back out, and when it goes to the heart again. Well, now we have low pressure, unoxygenated blood. It's going to return to the right atrium. So what we have here in this heart is RA stands for right atrium, LA for left atrium, RV for right ventricle, LV for left ventricle. So this unoxygenated low pressure blood is going to return to the right atrium, be then pumped into the right ventricle, where it's going to go out through some vessels towards the lungs. This is going to be, again, at high pressure. So this is high pressure, low oxygenated blood going to the lungs, where it's again going to go through a capillary bed and pick up oxygen. So it picks up oxygen through this capillary bed, and now we have oxygenated blood, but it's again at low pressure. So it's going to return to the heart through the left atrium and then go out again to the body. Now, the reason we have two circuits, the reason we just don't go from the lungs, essentially have a portal vessel or a portal system where it goes from the lungs into a portal vein directly into the body then, is that once this blood comes out of the lungs, because it's gone through a capillary bed, we are at low pressure now. And that's going to be a very inefficient way to deliver blood to the rest of the body. For instance, if you try to pick up your activity rate significantly, and you need lots of blood with lots of nutrients and oxygen delivered to your tissue very quickly, low pressure that's coming out of the lungs is really not going to cut it. You will probably pass out, something else like that. So we need to return this blood back to the heart so it can be pumped again under high pressure this time and pushed out to the body. So that's why we have these dual circuits in this our circulatory system, and that's why we have essentially this dual pump setup with the heart, is so that we can pump out to both of these two circuits. So let's look at what the heart would look like then, knowing this idea, kind of the anatomy of this, so that when we talk about these different parts later on, you have an idea of what's happening. Blood is going to be returning to the heart from the body through veins, particularly the superior and the inferior vena cava. It's going to come into the right atrium, and it's going to collect there until the right atrium contracts. Once the right atrium contracts, there's this valve that is between the right atrium and the right ventricle called the tricuspid valve, and it's going to remain closed as long as the pressure in the right ventricle exceeds the pressure in the right atrium. So the right atrium contracts, the pressure in this right atrium is going to increase until we open up the tricuspid valve and push the blood into the right ventricle. Once that happens, now we have this valve open. We are now going to contract the right ventricle, and it's going to push blood out. And again, the pulmonary vein is the pulmonary artery comes out of the right ventricle. And again, this pulmonary valve is going to remain closed as long as the pressure in the pulmonary artery exceeds that in the right ventricle. As soon as this happens, we build up pressure. We start contracting the right ventricle. Pressure is going to build up in this ventricle. It's going to close this tricuspid valve because now the pressure in the right ventricle is exceeding that in the right atrium. It's going to close this, but it's going to open the pulmonary valve because we have this increasing pressure here now, and push blood out into the pulmonary artery, where it's going to go to the lungs. Once it picks up oxygen in the lungs, it's going to come back by the pulmonary veins, return to the left atrium. Same thing essentially happens here, except for the valve that is between the left atrium and the left ventricle is the mitral valve. It's going to stay closed until as long as the left atrium pressure is below that of the left ventricle. But once it contracts, that exceeds that, push through here. The left ventricle is going to contract, close the mitral valve, open the aortic valve, push blood out towards the body through the aorta. So that's essentially the path of blood through the heart, and essentially the anatomy that we have in the heart. So as we start talking about this, more about the heart later on, we're going to come back and I'm going to talk about this anatomy. Just assume that you basically have an understanding of that from here on out. Okay, so let's talk a little bit about what's happening in the heart, and particularly the heart walls, are what we're going to be talking a lot about. So all the area that's surrounding those chambers. So we have a couple different layers here. We have the endocardium that has this layer of endothelial cells lining that. I'm not going to talk too much about that. We also have an epicardium, which is this thin exterior sheath that's lining the outside of the heart. But the really important one here that we need to talk about is the myocardium. This is a central portion of these heart walls, and it's composed of cardiac muscle fibers. This is the thickest layer by quite a bit because it's really doing the business of the heart. And these cardiac muscle fibers are arranged in this spiral pattern around the chambers of the heart. And what that means is that as we get contraction of these cardiac muscle fibers, it's going to essentially ring or pull around these chambers, and is going to aid in expelling as much of that blood as we possibly can. Now, not all of it's going to be expelled, but a good portion of it, and that's going to be aided by essentially this twisting as it compresses as well, and help expel a lot of that blood. Okay, so now that we're talking about cardiac muscle, let's talk about some of the characteristics of cardiac muscle and how that compares to both skeletal and smooth muscle. Well, first off, it's going to be striated like skeletal muscle was. So we're going to have the same myofibril arrangements that we had in skeletal muscle, and essentially all of that's going to be the same, and not like smooth muscle. we're going to have fibers that are unlike skeletal in that they're branching. So actually, instead of having one single muscle fiber that remains unbranched the entire distance, cardiac muscle will actually have multiple branching points through it. Now, cardiac muscle is completely oxidative. It's not glycolytic, really at all. It's one extreme end of the oxidative type of muscle. And as such, it has this really, really high concentration of mitochondria in the cardiac muscle, somewhere between 30 and 50%, depending on the species or the individual in that species. It's also going to have a very high amount of myoglobin. Now, if you remember, myoglobin is this oxygen-binding pigment that aids in pulling oxygen from the blood into the muscle cell. So as we have cardiac muscle that's very oxidative, it stands to reason that we also have this high amount of myoglobin to pull in that oxygen and hold on to it as well. Now, all these different cardiac muscles are going to be joined by this particular kind of joint between the cardiac muscles called intercalated discs. Now, they're essentially just this flat interface between the ends of two cardiac muscle fibers. And there's two different things in here that are important. First off are these gap junctions. Now, we talked about gap junctions before, particularly when we're talking about single unit smooth muscle, and then also when we're talking about electrical synapses in neurons. Now, in both of those, gap junctions do this really great thing that essentially we can connect the cytosol of two different cells, so the electrical activity in one gets transmitted directly into the cytosol, the next one, and we can pass electrical signals very easily between cells that are joined by gap junctions. We also have desmosomes in these intercalated discs as well, and these are essentially just structural adhesive proteins that keep that intercalated disc together and keep it during many periods of contracting from pulling apart at all. So the nice thing about these intercalated discs, because we have these gap junctions, is that action potentials in one cardiac muscle are going to very rapidly and very easily spread to adjoining cardiac muscle fibers as well. So we really have, once this starts in one cell, it's going to transmit to the rest of the cells that's connected to very easily. Now, one thing that this means is that unlike skeletal muscle, where we had this graded amount of contraction based on how many motor units we recruited, so the number of muscle fibers we could recruit to the contraction, that doesn't happen in cardiac muscle. It's essentially all of them or none of them are contracting, because since they're connected by all these gap junctions, once you start depolarizing, one of these cardiac muscle cells is going to spread to the rest of them very rapidly. Now, there is one kind of major exception to this, is that the atria and the ventricles are electrically separated through most of their contact. So there's only one point called the AV node, or the atrioventricular node, that we can get electrical signal from the atria into the ventricles. And so we have this control point there. But other than that, all these cardiac muscle cells are all connected through these gap junctions and these intercalated discs. So electrical activity is going to pass very easily between all of them. Okay, here's a little picture of a micrograph of cardiac muscle cell. You'll notice kind of up here that we have those kind of distinct striations that you can see also with skeletal muscle. But here in this blown up section, you can see here we have this intercalated disc. And this is unique to cardiac muscle. So now let's talk about the overall electrical activity of the heart. And really that needs to begin with the pacemaker cells and this pacemaker ability of the heart. So the heart is autorhythmic in that it can generate its own rhythmic depolarizations in and of itself. It does not need input from really the outside world, the outside system at all, like the nervous system. So in this way, it's somewhat like some single unit smooth muscle that we are talking about during the muscle section, in that it can generate this rhythmic pattern of depolarizations on its own. And this is going to be happening, again, like we had in smooth muscle, by non-contractile pacemaker cells. They're going to cause these rhythmic action potentials. Now, these are going to be primarily located in the sinoatrial node and the atrioventricular node of the heart. And we'll talk more about where those are in just a few minutes. And what this is known as is the myogenic electrical activity. So myogenic means originating in the muscle. So this electrical activity is originating in the muscle of the heart. Okay, so let's talk about how in the world does this actually happen. And to do that, we need to talk about how do we generate an action potential in these pacemaker cells. Well, turns out we have these particular sodium channels called the hyperpolarization activated cyclic nucleotide gated sodium channels, or HCN sodium channels. Now, the better name for them is just funny channels. They're called funny channels because they operate quite a bit differently than other voltage-gated sodium channels. And it turns out that they're actually open during hyperpolarization or polarization of the cell. So as these cardiac muscle cells become polarized, as you're coming to resting membrane potential, these funny channels are going to open. It's going to allow sodium to leak into the cell at a very high rate. High compared to what it normally does. And what this is going to do is it's going to gradually depolarize the cell. And these are all opening up right immediately after repolarization from the previous action potential. So as soon as we get done with the previous action potential, all these funny channels begin to open up and allow sodium to leak back into the cell and begin depolarizing it, bring it towards threshold. That's not the only thing that's happening. We also have potassium channels that are closing. So if you remember from neurons, what's primarily building up our resting potential is these calcium leak channels. Well, if we close those and we stop this sodium leak, this is going to aid in pulling us towards threshold potential, aid in this depolarization. But we also have something. As it gets closer, as the polarization or the depolarization gets closer to the threshold, we also have another kind of cell that opens up, these T-type calcium channels. And these open up just before we reach threshold, and we get this influx of calcium into the cell. So more positive ions flowing into the cell. And that's primarily because we have this greater concentration of calcium outside the cell, but also because we have an electrical gradient inside the cell, or pushing calcium inside the cell. And this further brings along depolarization. So we have these three different things that are aiding in this kind of gradual depolarization of the cell. Well, at threshold, what we have, and this is different, this is notably different than what happens in neurons, we have an opening up of L-type calcium channels. So, and it's going to cause a very large influx of calcium into these cardiac muscle cells. Now, this is important because it's not sodium that causes the rapid depolarization that you see during an action potential. So that very quick rise during an action potential in cardiac muscle is caused by a calcium influx, not a sodium influx. And then really what happens is we get repolarization kind of by the normal means. We have closing of the channels that were causing the depolarization in the first place, so the calcium channels close. We have opening of the potassium channels that allow potassium to rush out of the cell and repolarize the cell. And this is similar to neurons. So at that point, that's pretty similar. So this is kind of what you would expect to see over time with this rhythmic depolarization. As you see in this beginning, we have this kind of steady depolarization that's happening. And here at the beginning, this is initially happening because of funny cells. Or these funny channels, excuse me, not cells. And that is allowing sodium to leak into the cell, and we get this steady depolarization. However, over the last little bit of this, like right about here, what we have is the T-type calcium channels that are opening up and allowing calcium to come back in. That causes that. Now, this big rise that's happening right here, this is primarily happening because the L-type calcium channels are opening up and allowing calcium to rush into the cell. And then here, this repolarization is again because of these L-type calcium channels closing and potassium channels opening up right there. Potassium rushing out the cell. So this action potential is a bit different than the action potentials that we've been seeing before. There's different ions at work that are causing it, but really the gist is the same. The big thing here is that these funny channels, primarily, but also the T-type calcium channels, are leaking in positive ions and bringing the membrane potential towards depolarization. So what happens is we can action potential, repolarization, and gradual depolarization to get it back to this. And this is the process that is setting the rate and the pumping of your heart, the rate at which that beats, and setting that beat. Now there's regulation that comes in and alters this and changes that rate, but this is the fundamental process that's causing your heart to beat. Okay, so let's talk about the spread of this electrical activity. Once we produce this spontaneous rhythmic depolarization, what happens after that? And really, all of this is going to begin in the sinoatrial node. Now, both the SA node and the AV node are both autorhythmic. They're both generating these kind of rhythmic depolarizations. However, that being said, the SA node, this is happening faster. And the AV node, since it's electrically connected to the SA node, when it depolarizes, is going to cause, eventually, the AV node to have an action potential as well. And so it's really the SA node that ends up controlling the pace at which this happens. So the SA node is setting the pace, or at least the inherent pace for the heart. Now, signal is going to travel through these specialized conduction pathways of more of this kind of autorhythmic pacemaker cell type cells. Now, one thing I need to point out here, however, is that even though these are autorhythmic as well by themselves, they're not actually providing any rhythmicity, kind of like how the AV node isn't. They're taking their depolarizations or their rhythm from the SA node. But what happens is once the AV node fires, because these other specialized cells, these other autorhythmic cells, pacemaker cells, have been drifting towards depolarization, or drifting towards threshold, they're closer to threshold when that signal comes around than the rest of the cardiac muscle is, which means that they will depolar, or they will reach threshold quicker, which means that we essentially have these cells running throughout the heart that will depolarize more quickly, or come to threshold more quickly, than the rest of the cardiac muscle cells. And those provide this very quick transduction of that signal throughout the heart before the rest of the cardiac muscle contracts. And so these act as kind of in the same way that neurons do, that they're going to pick up and carry this electrical activity through the heart and kind of transmit these action potentials and help coordinate the contraction of all the cardiac muscle together at the same time. Now, what's going to happen is first, after the AV node, or the SA node creates an action potential, some of these conduction pathways, some of these fibers are going to carry that action potential throughout the atria, and the atria are all going to depolarize and contract. That's the first thing that happens after the SA node reaches action potential. Also, the signal is going to reach the AV node as well. So there's this specialized conduction pathway straight to the AV node. But at the AV node, there's going to be a delay in this signal for about 1/10 of a second. It's going to hold up right there. This is going to allow the atria to completely contract before we start contracting the ventricles. Because we don't want those contracting too early. They need to receive all the blood that's being squeezed out of the atria before we begin to contract the ventricles. And after this delay, the signal is going to be propagated through more of these specialized conduction pathways, particularly through the bundle of His, which is a large conduction pathway immediately off the AV node, and then these paired conduction pathways down the septa that separates the two ventricles, called the Purkinje fibers, that then curl around, innervate the rest of the heart muscle, and carry these action potentials throughout that. And this is going to cause all the ventricle myocardium to contract at the same time. It's going to coordinate that contraction. So let's take a look at how this works here. So we look over here at this image on the left side. This is coming from your textbook. Up here in the right atria is the right atrium is the SA node. This is where our signal is going to be originated. And we have these conduction pathways. We have our AV node here. So it's going to first show up here, go throughout these conduction pathways, and cause a contraction of both of these atria simultaneously. All the while, this action potential is going to move to the AV node, where it's going to be delayed about 1/10 of a second, allowing these atria to fully contract, and then being sent down these Purkinje fibers, or down the bundle of His, down the Purkinje fibers throughout the rest of the ventricle myocardium, and then coordinate those to contract at the same time as well. So that is how this is going to be propagated throughout the heart. Okay, now let's talk about, now that we've kind of got, how does this spread? How do these pacemaker cells work, and how do they propagate this signal and generate this signal? How do specific cardiac muscle cells, how action potentials in those operate as well? And again, this is going to be slightly different than what we saw in neurons, what we saw in skeletal muscle, and what we even saw in the pacemaker cells of cardiac muscle. Now, the rising phase, unlike the pacemaker cells, is going to be due to sodium channels again, not calcium channels like we saw in the pacemaker cells. And after this happens, these are going to be inactivated after they go through an action potential. These sodium channels are going to be inactivated for a very long time, about 250 milliseconds. And that means that there's no summation possible or no tetanus possible in cardiac muscle. These sodium channels that begin the action potentials do not reset fast enough to allow for summation. Now that's a really good thing, because when you start like your heart beating really fast, you don't want to have summation, essentially just your heart going to tetanus. You don't want just to clamp down and not relax again. And so this is a really important thing for the operation of cardiac muscle. So after we have our rising phase that occurs normally due to sodium channels opening, we then have another normal thing that happens, potassium channels that open, produce repolarization. However, these potassium channels only open transiently. They're only going to open for a very short amount of time. It's not going to completely repolarize the cell, just partially repolarize the cell. Then those calcium channels are going to cut out, and we're going to have calcium channels that are opening up. These are more L-type calcium channels. And this is going to produce a plateau, because now we have calcium that's pushing into the cell, and it's essentially going to hold the depolarization stable or only barely repolarizing for a period of time. Now what this does is this is going to lengthen the action potential, somewhere in the neighborhood of about 100-fold lengthening of the action potential compared to what we would see in something like a neuron. This is going to last somewhere between 250 to 300 milliseconds. And what this does is this allows for a longer, more powerful contraction of the cardiac muscle. The way we're doing this, we don't want to have summation, so we need to have a single twitch that is strong enough to drive the blood out of the ventricle or out of the atria and into the vessels that it's going into. And so this needs to be a longer action potential that is able to drive the fluid onto its next stop. And so we do that by lengthening this action potential and allow for a longer, more strong contraction of the cardiac muscle. Now after that happens, again, we finish this off like we do most action potentials. We close down the channels that were open, in this case the L-type calcium channels. And then we have these different calcium channels, not the ones that were transiently opening before, but a different class of them, opening back up allowing potassium to rush back into the cardiac muscle cell and repolarize that cell. So let's look at what happens here. This is what it looks like. What we have is the rising phase here that is again caused by sodium channels opening up. We have this transient repolarization that is caused by potassium channels opening up and potassium leaving the cell. But then we have this plateau phase right here. What's happening here is we have our calcium channels opening, calcium entering in the cell, and remember these are L-type calcium channels, they're long-lasting type calcium channels. And that's going to hold essentially, it's going to be slightly repolarizing, but essentially hold this action potential in this depolarized state for a little bit of time. And then we have, again, potassium channels opening up again here, causing a rush in and this rapid repolarization. So this is the look of a heart or a cardiac muscle, a contractile cardiac muscle action potential. It's a little bit different. We have a tweak on the theme, namely this rushing in of calcium that prolongs the action potential, causes this plateau phase. Okay, so as this happens, like we saw in this plateau phase, it does a couple other things. Not only does it lengthen it, but it allows calcium entry into the heart muscle, or into the heart muscle cell, and aids in contraction. And we also have this action potential that's going to travel down T-tubules, like we saw in the skeletal muscle, and again, this is going to open up calcium channels. Now this is going to operate slightly differently than what we had in skeletal muscle. Remember, the calcium channels, the T-tubules, were mechanically adhered to calcium channels in the sarcoplasmic reticulum. That's not going to happen here. What we have actually is the T-tubules have their calcium channels that open up and allow calcium into the muscle cell all the way down, and that calcium that's entering from the extracellular fluid into the cytosol through the T-tubule calcium channels is then going to activate sarcoplasmic reticulum calcium channels to open and also allow calcium to come in from that. So that is where we're actually kind of getting this combination of both skeletal muscle function and smooth muscle function, that we're both getting calcium in pretty significant quantities of calcium, both from the extracellular fluid and from the sarcoplasmic reticulum. So that's how this is kind of slightly different than both of those. I guess I already mentioned this, the calcium rushing into the cytosol from the ECF. We're going to open up the sarcoplasmic reticulum by activation by calcium, not by this mechanically adhering of the calcium channels in the T-tubule to the sarcoplasmic reticulum. Okay, that was basically the operation, the electrical activity, the electrical operation of the heart. Let's look a little bit at how this gets measured in actual organisms like humans. And you've probably heard of this, an electrocardiogram, which is abbreviated EKG or sometimes EKG, which I've both heard because it refers to the Greek cardio, which is spelled with a K, and sometimes to the German cardio, which is also spelled with a K, because I believe it was actually a German that ended up doing most of the pioneering work with EKGs, a guy named Willem Einthoven. Anyhow, kind of beside the point, what an electrocardiogram is when you have a depolarization of your heart and all these cardiac muscle cells in your heart, this is going to cause an electrical current that actually travels a good ways throughout all of your body and shows up on the surface of your skin. And we can actually measure the voltage that is produced on your skin by the depolarization of these muscle cells on your heart, and that is an electrocardiogram. We can get a good idea of what's happening with your heart measuring this voltage current or this electrical current that's winding up, being conducted to your skin surface from your heart. Now, a couple things to keep in mind here, though, is that whatever is happening, this is a summation of all the combined electrical activity of the heart combined. So some things get obscured and some things don't really show up. So, for instance, the actual operation of the SA node and the AV node, that's too weak to actually show up on EKG, and some things occur at the same time, so they don't show up there as well. Okay, so let's talk about the individual components of this. This is what you would see for a normal, clean EKG. And you have several different components here. I'm going to label those off. But first off, you have this first bump right here. And again, this is time this way. And this would be voltage going this direction. So essentially a graph. What we have here, this very initial deflection, is called the P wave. And then we have these three different points: a downward deflection, a very sharp upward deflection, and then another downward deflection. Each of these points has their own letter as well. This is Q, this is R, and this is S. Together, this whole section right here is called the QRS complex. And then finally, this last deflection right here is known as the T wave. So really, it's just this clump of letters from the middle of the alphabet, P, Q, R, S, T. So, that's what they're named. Let's talk about what actually they are. The P wave, we're over here, this is your atrial, or your atrium's depolarizing. That's the very first thing that it's a very initial, large electrical thing that's going to happen during the course of your heart contraction. During the QRS complex, really what you're seeing here is ventricle depolarization. So this is ventricle depolarization. During this time, atrial repolarization, all that cardiac muscle in your atria is repolarizing in here as well. This is essentially covered up or summed up in this ventricle depolarization. Now this is much bigger than the P wave. The QRS complex is much bigger than the P wave because there is a lot more muscle in the ventricles, and so as they contract, as they all have action potential simultaneously, that electrical signal or that electrical current is much bigger because we're summing up all of those muscle fibers compared to the atrium, the atria, which have a lot less muscle. Finally, the T wave is ventricle repolarization. And so here, all the ventricles are repolarizing, relaxing, and that's what you have happen here. Well, let's imagine here just for a second we could actually look at action potentials happening in individual muscle cells, one of the atria and one of the ventricles, as this is happening. This is essentially what you would see happening here. This is the atria. We're looking at action potentials here. This is in the ventricles. So what you have here, right here at the beginning of the P wave, we begin to have the depolarization, or this rising phase in the atria. So this right here is caused primarily by sodium rushing into all the atrial muscle fibers, and that is why we have this initial deflection right here. Here in the QRS complex, we have a couple things going. The main thing that you're seeing here is the ventricle depolarization right here. Now, one thing I need to say is, during this segment here, which is called the PR segment right here, the atria are contracted this whole time. This is the time in which the atria are contracted. At the QRS complex, we get relaxation of the atria. This depolarization stops. And we get this influx of potassium back into these muscle cells here. But the big thing that's happening is we have this influx of sodium into the ventricles, into the ventricle muscle cells, not into the ventricles themselves, and causing a rapid depolarization. And that's what causes most of this electrical activity right here. At the end of that, then, we have this time in which we have, after this action potential finalizes, we have potassium rushing back into, out of the ventricle muscle cells. That causes this deflection here, the T wave, and that's relaxation of the ventricles. So really what happens in here is the ventricles are contracted. And so that's essentially what's happening with the electrocardiogram. Okay, when we pick this back up after next class period, we're going to be talking some more about the actual mechanics of the heart, moving on from electrical activity to talk more about macro mechanics, and then moving on to the rest of the components of the cardiovascular system.